Hello students, welcome back to my biology online classes. Today I am going to discuss with you about another type of cell surface receptor, a second major type of cell surface receptor. That one is enzyme linked receptors, the first major types of cell receptor family. GPCR I have already discussed with you in detail in my previous lectures. Today I am going to focus on this topic, enzyme linked receptor. As the name suggests, enzyme linked means there must be an enzyme related to perform all these phenomena. They are also like GPCR, they are also transmembrane. Obviously they are protein in nature. Ligand binding domain is on outside of the cell. Ligand binding domain, this is outside the cell, this is ligand binding domain, and this is cytosolic domain. This one is cytosolic domain, ligand binding domain. Cytosolic, so two domain, ligand binding domain and cytosolic domain. Write it in their full form. Okay, I am not going to write ligand binding domain and cytosolic domain. And in this cytosolic domain, they are cytosolic domain, CED. Cytosolic domain, this cytosolic domain has an intrinsic enzymatic activity or associates directly with an enzyme. In case of GPCR, what have we seen? That their cytosolic domain associated with a monomeric G protein, but in case of this enzyme link, inside their cytosolic domain which remain inside the cell, it has either an intrinsic enzymatic activity or it associates directly with an enzyme. Somehow it's related to enzyme, and several classes of enzyme linked receptor have been identified, and I have already discussed all the types, just type and example in my previous lectures. Now let's have a look. The types are receptor tyrosine kinase, RTK, which is in your syllabus, and this is the RTK. This is the most numerous type of enzyme linked receptor and very much important. Point number 2, tyrosine kinase associated receptor. Point number 3, receptor like tyrosine phosphatases, receptor serine threonine kinases, and receptor guanylyl cyclases, but these are not in our syllabus. Tyrosine phosphatase, serine threonine kinase and guanylyl cyclase, these are not in our syllabus. So we are going to study, concentrate about RTK, receptor tyrosine kinase and tyrosine kinase associated receptor. Now let's concentrate about RTK, receptor tyrosine kinase. Receptor means this one is cell surface receptor, common knowledge. Tyrosine means amino acid residue, all we have know, and kinase enzyme means it add phosphate group and it is an enzyme and protein in nature. This is second major type of cell surface receptor, and first major type of cell surface receptor is GPCR. I have already discussed it. And what are the ligands for RTK? Ligands means signaling molecules that will come outside of cell from an excited cell, and that cell receives signal from its surrounding and being excited, and after being excited they release these signaling molecules or ligands. So ligands for this RTK, okay, insulin, epidermal growth factor, EGF, TGF alpha, transforming growth factor alpha, PDGF, platelet derived growth factor, FGF, fibroblast growth factor, CSF1, colony stimulating factor. These are all about ligands of RTK, domains of RTK. All RTKs have extracellular domain and cytosolic domain. Extracellular domain. Ligand binding site. A single hydrophobic transmembrane alpha helix, a cytosolic domain. This is inner side, inner. Okay, and here there is a region with protein, tyrosine, kinase activity. So these are domains of a RTK or receptor tyrosine kinase. Ligand binding site, single hydrophobic transmembrane alpha helix. Hydrophobic means it rejects water. Transmembrane means membrane, here membrane, transmembrane, some part outside membrane and inside cytosol. Okay, and they are cytosolic domain, which is inner side, and there is a region with protein, tyrosine, kinase activity. Most RTKs are monomeric in nature. Binding to the extracellular domain induces formation of receptor dimers. Very much important. Receptor dimers. Normally they are monomeric, means single, and after ligand binding to extracellular domain, the changes occur inside, means in cytosolic domain, and two monomers become united to make a dimer. This one is very much important, and mind it very carefully. Normally they are monomeric in nature. After binding ligand at the extracellular domain, there is a change and two come to the close contact and make a dimer for response to ligand binding. And after this ligand binding, what happens? Activation of intrinsic protein, tyrosine, kinase activity of receptor, autophosphorylation of tyrosine residues. As tyrosine kinase, autophosphorylation occurs at tyrosine residues, and it occurs in its cytosolic domain. Is it very clear? Normally they are monomeric. After binding ligand at the extracellular ligand binding domain, there are changes occurs in cytosolic domain and two monomer become united to make a dimer, and after formation the dimer induces, what induces? Activation of intrinsic protein, tyrosine kinase activity of receptor, and their autophosphorylation of tyrosine residues occur in the cytosolic domain. Now receptor is activated and this can now phosphorylate other protein substrate. They can now phosphorylate other protein substrate. Now let's have a look about the diagram. Along with this theoretical portion, you should mention about diagram. Without that diagram and mention diagram, the description of RTK will remain incomplete. So let's concentrate about diagram. When you are going to draw it like this: ELP and protein, lipid, protein layer. This is one monomer. Ligands. Normally this extracellular domain, this is cytosolic domain. This is one monomer, and here there is also another monomer, transmembrane. Both of these are this and this kinase catalytic site. This one and this one is ligand binding site. Yeah, ligand binding domain. ATP to ADP. Release of one single phosphate plus energy. They come in close contact. Ligand binding domain. In ligand binding domain, ligand binding is done. Both of these are ligand. Both of these are phosphorylated and forming a dimer. They can phosphorylate each other, and the place, and this place is tyrosine residue. Okay, phosphorylate in which place? At tyrosine residues. The activated kinase, they now can phosphorylate another, and the process is going on. Okay, activation of RTK is done. The resultant phosphotyrosine functions as a docking site for many signal transduction. So this is all about RTKs.